I'm James, so I'm from Stanford. So I'll tell you about a couple of the recent projects that we've been working on. So the main talk will be around how to quantify the value of data, especially in biomedical context. So my group works on quite a wide range of applied machine learning problems. So we develop machine learning algorithms for largely motivated by biotech and more recently by medical clinical applications. So I actually changed the talk a little bit, also based on some of the interesting things I saw from the morning talks. So instead of, before jumping into the data evaluation parts, I actually want to give you sort of a concrete example of one recent applied ML work that we've been doing, so applied machine learning to improve genome editing. And then I'll talk about, given that context, talk about the value of the data in machine learning, especially in biomedical applications. And then inspired by Casey's talk from the morning, so I'll actually tell you about a new platform that we developed that makes it extremely easy to share machine learning models, especially with biologists, clinicians, and non-experts. It's called Gradio. OK, so we'll start with genome editing, and feel free to stop me if you have questions. Right, so many of you have heard about all this excitement around genome editing. It's probably the only thing right now that's getting more hype than AI, right? More excitement. So there's also a lot of uncertainties around both the ethical questions throughout editing in humans, right? And very much intertwined with that is sort of the safety questions, right? So if you actually make these edits in the human cells on human DNA, so what are the potential unexpected outcomes, right? And there's been a lot of controversies around both of these questions, and many of you maybe have read about earlier this year of this editing in China in humans. So what I want to do today is to show you an example of how we can use machine learning to actually try to improve the safety of some of these genome editing process. Right, so before getting into that, since this is more of a tutorial, so I just want to bring everyone up to speed on how this editing process works. Right, so this is a very quick primer of the editing. Right, so if you think of the editor, it's basically this enzyme, right? So this little molecular scissors, right? So it goes into the nucleus, and then you can design it to bind onto any particular location in the genome that you're interested in, right? Or most locations. So it goes to that location, right? And now it latches onto that location like zippers, right? And here's the interesting part, the important part. It actually creates a double-stranded break at a very precise point, right? And now the genome's broken, right? So it tries to do this random walk to try to find the right combination of nucleotides to stitch itself back together again. Right, so it's just very much a random walk before it finds the right combination. And so the last part of this random walk, that's actually where a lot of the uncertainties and safety concerns comes in. Right? Because it's a stochastic process, you can't really control exactly what gets inserted and deleted. And that's what we want to model and to understand the stochastic process. So there's a couple of ways you can frame this problem. Right? So there's the machine learning framework, which is if I won't tell you which location you want to make the edit, with the cut, so predict for me what are the likely insertions, deletions, and substitutions, mutations that likely happen at that location. Right, so that's sort of the ML prediction framework. There's sort of a complementary statistical modeling framework, which is given all of the data you have, right, can you actually use this data to infer processes about the DNA repair process, right? Maybe that can inform the underlying biology. So we've been working on both approaches, but I'll tell you more about the first one, the ML approach, since that sort of fits in more closely with the rest of the talk. And this work was actually just published on Monday this week in Nature Biotech, describing these algorithms that we have. So basically, the idea here is that we are going to generate a very large data set, right? One of the largest data sets in primary human cells in order to model and to train this process, to understand this process. So before I dive into what we did, so I want to tell you about sort of a general conceptual landscape of what are the computational problems we are involved around genome editing. Because I think this is a place where a lot of you can make important contributions. Still quite open space. So in my mind, there are basically three categories of problems, computational problems for genome editing. Right? So this is the on-target effects, which is what we study here. So that means that if I actually make a cut at a particular location, at that location, on that target, what is the probability of insertions and deletions and mutations at that location? So that's our focus. There's similarly an important question about off-target effects, which means that if I want to target a particular part of your chromosome, but maybe the enzyme goes to other locations, other genome, and there are other chromosomes, right? So what are these unexpected off-target locations? Lots of interesting computational methods, interesting questions. Right, the third bucket is when you actually apply these CRISPR edits to large, for example, large screens, all these other variants, right? So how do you actually analyze that kinds of data for downstream biological interpretations? Also really important. So in order to address the on-target effects, so we've generated in collaboration with the Chan Zuckerberg BioHub, I think one of the largest data sets in primary cells, human cells, of these on-target CRISPR edits. So let me tell you briefly about the data. So what we do is that we actually take primary human T cells, right? We'll go across the genome in about 1,500 different locations. Right? At each location, we'll make the cut. Right? We'll make the cut, and then we do deep sequencing at the location to say, OK, so in this population of millions of cells, how many of them will actually have insertions and deletions at that location where I make the cut? Right? So that gives me a set of observations, a very rich set of observations about this repair process at that location. Right, so the data that we collect will have basically the following forms. This is an example of it. Right? If I make a cut at this location, each row of this corresponds to basically one type of repair outcome. Right? The numbers here corresponds to how many reads I have, which somehow indicates how likely is this outcome to happen in some subset of cells. Right? You can see that, OK, so at this particular location, maybe I'll get some short deletions. Other times I get longer deletions at different frequencies across the cell population. And we do this across these 1,500 locations in T cells. And this is repeated across actually 18 different human individuals, right, to see what are the genetic variations. And then we repeat this whole thing again across different cell types, including iPS cells and other cell lines. So it's I think quite a nice tensor of data sets that we collected for this. Right? And the most important part is that for every experiment, we actually get 60,000 reads. So you can think of each read as being an ID sample from this distribution of repair outcomes at that location. Right, so does the problem and data set make sense? Yeah.
You have also insertions or only deletions?
You also have insertions, yes. Yes, actually, the insertions actually become very interesting, so I'll tell you where the insertions come from. And that's actually, in some sense, the biggest surprise in our study. So the idea here is that we want to, since we're framing this, taking this supervised machine learning perspective, right? So the formulations that I want to take as inputs, my DNA sequence, maybe other physics and chemistry on this DNA sequence. I want to predict for you a set of outcomes that will give me some indication of how safe is it to make a cut at this location. Right? So the outcomes I want to predict are, if I make a cut here, how likely am I to get insertions versus deletions? Right? If I get an insertion, how long it's likely to be, or how long are the deletions likely to be? And overall, I want to get some sense of what is the entropy of this process at this particular location, right? Because I want to prioritize perhaps regions that have low entropy or higher precision. So we developed a variety of models to make these predictions. I'll tell you a little bit about some of them. So one type of model that you might try to apply here would be sort of a more standard kinds of supervised or convolutional neural networks, right? So you have this DNA sequence. You can view this DNA sequence as sort of a one-dimensional genome, right? A one-dimensional image. And then you can basically scan little filters across the nearby DNA sequence to predict this set of outcomes. Right? So that actually works reasonably well, but I'll tell you something that ends up, I think, is potentially a much more powerful approach. Right? So instead of representing the DNA as this one-dimensional ACTG, one-dimensional image, we're going to view this as a two-dimensional image, this two-dimensional dot map. Right? So how we generate this two-dimensional image is I just take my ACTG sequence at the cut site, and I just align it against itself. Right? I have a black here if it's if they have the same sequence, and white otherwise. Right? So you have these blacks along the diagonal, as you expect, and you also have these stripes, right, that are going slightly off the diagonal, which corresponds to microhomologies. Right? So these are sequences that are, in this case, this TCGA, right, that's repeated at different locations near my cut site. And the reason why this is useful is that we know that these microhomologies, or short repeated sequences, are actually very important in driving the different kinds of DNA repair process, for example, in inducing insertions and different types of deletions. So we want to explicitly capture them. Right? So if we do this right across this entire cut site, and then you generate this kind of carpet-like pattern where you have all these stripes, and they'll tell you quite a lot of interesting information about the different microhomologies and the pattern around the genome. Right? And then we basically scan across this two-dimensional carpet of this two-dimensional DNA dot map to make the predictions. Okay, so in addition to just making the predictions, right, so if we really want to make this a useful and reliable tool for, especially for therapeutic applications, in really real human cells, we want to be extremely rigorous in how we evaluate and test this model, right? More rigorous than perhaps the standard ways to test machine learning models. So how do we do this, right? So the first set of tests is basically what people typically do in supervised ML, right? So you leave out some chromosome, you leave out part of the chromosome, and you can make these predictions. And here we're talking about three types of predictions, right? So for precision, right, how precise is the edit outcome at each location, right? How likely is it to have frame shift? And the part that's hidden here is basically how likely are you to have insertions rather than deletions. So Sprout is the algorithm that we developed, right? So it's getting, I think, quite reasonable accuracies on these T cells. There's actually a lot of intrinsic biological variation here, so we're not going to be able to expect to get close to 100% accuracy, but it's getting, I think, quite relatively good accuracies. These are on the test T cells. Right? And it's doing significantly better than a couple of other recently developed methods, also trying to make these similar kinds of predictions. But this is done on T cells, right? This is test data, right, from this data set that we generated. So then what we did is in order to really be sure, is that we went back after the algorithm has been developed. We actually collected new data from new human donors and testing at different locations that has not been seen before, right? In different lab, new individuals, new locations, just to see really how robust the model is. And it's actually quite reassuring for us that, okay, so the model performance is essentially the same as before, right? So it's really actually doing quite well. And then we actually want to go one step further to really stress test it, right? So then we went back again for the same model, the model is fixed, right? We went back and collected another set of new individuals, but this time in a different cell type, right? So iPS cells rather than T cells, and also in new locations, right? So this is basically as different as we can get, new individuals, new experiment, new cell type, new locations. The model has only been trained on the T cells, right? So you can see a relatively actually stable performance even across these new cell types, which give us quite strong confidence that the models actually is quite robust even to different settings. And we've done a lot of other validations. You can also do various interpretation methods to interpret that the presence of actually particular nucleotides, for example, the presence of a G here next to the cut site, actually really makes it more likely to have deletions, and presence of A here makes it more likely to have insertions, right? That gives us actually mechanistic insights about the DNA repair process. Okay, so far, so good. But let me tell you something that's actually quite surprising to us, right? And that's comes back to these insertions. So the textbook picture here is that, again, you have this random walk, right? So the insertions, deletions, right, really should be fairly local, right? Maybe you have 1 or 3 nucleotides change, but you don't really expect large deviations from that because it's a local Markov chain or random walk. What we did observe is that actually in quite a large number of locations, in over 90% of the targets cut sites, we observed actually quite long insertions, right? So longer than 25 nucleotides, so much longer than you expect from this local random walk. And these long insertions actually come from quite interesting other locations in the genome, in the chromosomes. Right, so we looked quite carefully to see where these long insertions come from. Right, so here, just to be clear, we're not actually putting in any DNA templates. Right, so there's nothing we're explicitly trying to insert. So these insertions are really just happening organically due to the DNA repair process. So it turns out that actually a lot of these long insertions we think actually come from other chromosomes, but other chromosomes that are in three-dimensional contact with the location where we actually make the double-stranded DNA break. A quick way to visualize this is that, okay, so on the left here I have it's kind of, you get these three-dimensional contact maps between different chromosomes using high C. Right? On the right here I have basically how likely is one chromosome to be, part of it to be inserted into another chromosome, right, if CRISPR is targeting the second chromosome. Right? If CRISPR is targeting chromosome 1, how likely is chromosome 2 to be partly to be inserted into that cut site? You can see there's actually quite large similarities between, even visually, between the contact, probably a physical contact, and these cut sites. Right? So our mental model is that as the DNA is being cut, right? So it's basically trying to find the right ways to stitch itself back together, and it's essentially looking for other DNA sequences that are physically nearby. Right? So even if it's double-stranded DNA from other chromosomes, there's some chance for that actually to be inserted. And we've done a lot of other analysis and validations in T cells and different cell types to show that it's actually really likely to be, it's really like the physical contact due to three-dimensional interaction that's driving these insertion events. Right? And I think that's actually has quite important implications for if we're really trying to apply these tools for CRISPR editing tools for therapeutic applications. Right? So even though these long insertions are relatively rare, they happen in, by our estimate, in less than 1 in 100 cells. But they are quite prevalent, right, across different parts of the genome. And the parts that are getting inserted are oftentimes different enhancers or open chromatin regions because they're physically active and available. Right? So we don't, so it's really important to understand what are getting inserted when we make these edits. So we've made this tool Sprout that's available, and so people can put in your own sequences, and we'll make these predictions until tell you what are the likely insertions and deletions. Okay, cool. So any questions about this part before I move on? Good. So this is kind of an illustration, right, of how we typically do sort of these applied machine learning projects in biomedical applications. Right? We go out, design the studies, collect these large data sets as training data, and we'll collect additional data as validation data. And in this case, the data we collected with help of the Chandra BioHub, which put in a lot of resources here. Right? So the data are quite expensive. The experiments are really expensive. And this leads to a general question of, if we're collecting these data from all these different sources, especially for biomedical applications, right, how do we actually really quantify the value of different experiments and different types of data that I'm collecting? Right, so this is another example of a very widely used data set from the UK Biobank. Right, so the UK Biobank, actually data come from these 22 locations in the UK. And here I'm showing you the number of participants from each of these locations. Right, so for example, it's natural to ask, OK, so in the UK Biobank, how much value actually, right, is being contributed by each of these locations, right, or by each of the individuals into your data set. So beyond, I think, very important scientific applications, right, if we can figure out the value of different types of data, then we design better data sets. We can better optimize for how we collect for data. I think this is also really drives the heart of a lot of recent policy debates. So even just a couple of weeks ago, the Dashboard Act in the US Senate would propose that large companies that are using consumer data in the US will have to quantify the value of the data that they collect, right, for both regulation and taxation purposes. Right? So they have to quantify, come up with a value for that. And locally in California, right, so the governor of California, Governor Newsom, has discussed his proposals for data dividend, whereby individuals in California would also be compensated by the large companies, Google and Facebook, for example, for the digital data that individuals are generating in California. Right? So all of these proposals are very interesting policy proposals, right? And they're just going to become increasingly prevalent as we go forward. But the fundamental question here is that it's never really clearly defined what does the value actually mean of the data, and how do we operationally quantify that. Right? So we think of this value of data, especially as more and more applied ML applications are being used in deployment. Right? It's being sort of the core question that really drives a lot of the problems that we would like to solve going forward, right? Both for experimental design purposes, but also for these broader policy questions. And if you think about it, it's actually quite challenging, right? Because the modern kind of applied ML framework, for example, the one I was telling you about for CRISPR, is that you take this data from different experiments, or maybe from different hospitals or different individuals, and the data is being intertwined in some horribly complex nonlinear black box. Right? So then how do you actually figure out what is the value of each hospital's data or each individual's contribution to that, to the whole mess? So we've been quite interested in this question. Right? We've been, especially for the biomedical applications. And basically, the first thing that we want to do is actually define more precisely what exactly does it mean to quantify the value of data. And then we want to design sort of efficient algorithms to compute these values. And we're also working closely with the policymakers as well as the companies like Google to deploy these methods. Right? So this is sort of a step towards a broader framework of data governance that I'm quite excited about. And we have a line of recent research, ongoing research to address these problems. So for the talk today, I just want to look at one small concrete piece of this, which is going back to these kind of supervised applied ML problems. How do we quantify the value of different types of data in a supervised machine learning algorithm? Right. So just to be very precise, right? So there are basically 3 ingredients of a standard supervised learning algorithm. Right. So I have some training data, which could be my different CRISPR experiments. Right. I have a particular learning algorithm, right? It could be my ConvNet, or it could be an SVM trained on these CRISPR data, right? And I achieve some performance that I care about, right? You tell me what performance you care about. It could be, the F1 score or accuracy of my predictions, right? So, and that's my, and maybe on this particular data set, I achieve, like 80% accuracy. So my goals here, very concrete, is to say, okay, so how much did each of these individual data sources or data points, right, contribute to that 80% accuracy? Right, so I would like to basically partition that 80% accuracy to each of my sources of data. Right. So that's in a way that's sort of reasonable. Right. And this is where it becomes more complex, right? So there are potentially infinitely many ways you can try to partition the performance of your model back to each of the original data points. Right. So how do we think about, what's a good way to do it? For example, one way you might think about doing this is just to do like a leave one out kind of estimate, right? So you just take a data point out and see how does that leaving that out change the performance of the model, right? So that's actually what commonly used in statistics and machine learning by the name of leverage score or influence scores, right? Maybe is that a good way to do it, or are there other ways to do it? So how do we even think about this framework in a more principled manner? So we've been borrowing on these ideas. It's actually been quite widely used in economics, right, of what are called Shapley values. So the idea here is it's quite simple, right? So it's the following setting. So imagine if all of us in this room, right, if we work together to solve some task, and we get a bonus in the end because we've done well, right? So how do I actually split that bonus among each of you so that everyone is happy with your share of the bonus? Right, so and nobody complains. Right, so in economics, it's a well-defined framework for how to do this. And it turns out that under very general conditions, there's a unique way for doing this. So in our setting, so I think of instead of each of us working together, imagine now you bring your own data set. Right, so it's the data that works together to train my machine learning model. So the bonus I get in the end is the performance of my model achieves. Right, I would like to split that performance among each of my data points. Right, so that's what we call this sort of extension, the data Shapley value. So there's a set of very general conditions under which you can actually show that this Shapley value is the unique way to split up, to value data sets, right, that satisfies very general conditions. Right, so it's very briefly, in these conditions, the first one basically says that if you have a particular data point, if adding that data point to a training set, right, never changes the performance of that training set, you should assign 0 value to that point. Right, the second one just says that if 2 data points always contribute the same, changes the data sets in the same way, they're like twins or interchangeable, they should be given the same value. And the third one basically says that in machine learning, we're typically making predictions over a large test set. Right, if I add the test point to that test set, then I should just simply modify the values by the corresponding contribution for that new test point. And it turns out that this is what was proved in the game theory literature, right? And we can extend that to the data setting. That the only way to satisfy these properties is given by the following fairly intuitive formulation. Right, so let's parse this. Right, so basically, if I want to compute the value of a particular data point, right, in some statistical model or in some machine learning context, so what I do is that I would look at different subsets of the data set, maybe subsets of size 1, 10, 100. For each subset, it's called that S here, I'll add that data point to it and see what is the change in the performance of my model when I add this data point to that subset. Right, so it's the marginal contribution of the data point to different subsets. Right, and there's a particular simple normalization term. So you can basically view this as sort of like expected contribution over different types of, over different subsets, subsamples of my training data. So like a simple, quick illustration of this, right? So if I want to compute the value of star, right? So I would train my model on star, and I would remove it, and I see how much the performance change. But then I also look at different subsets, right? And then also remove the star and see for each of the subsets how much did my algorithm's performance change, right? And you can end up so you can do this to and aggregate this together to get this data Shapley value, right? And that's the unique formulation that satisfies these reasonable properties. Let's see. OK, so let me actually show you this part, right? So going back to this UK ballot bank data in the beginning, right? So I said, OK, so now we can actually apply this formulation to compute what is the contribution of each of my locations, right? These 22 locations, right? So and here we do this for particular tasks, right? So let's say we're trying to build a model to predict or to diagnose lung cancer. Right, for that task, I'll take the data from the UK ballot bank, the genotypes and behavior lifestyle information from each of the individuals, and build a predictor for that. Right, and then we can basically apply this formulation we have to compute the data Shapley values. So here, basically in blue, I'm plotting for you the value from each of these locations, right, each of the 22 locations that we can actually compute. What's quite interesting, a little bit surprising to us, is just how heterogeneous these values are. Right, so in particular, you have locations like that 15 there, which is Nottingham, where you actually have negative values, which is mathematically possible. Right? And having a negative value actually means that somehow like the data from that location is hurting the performance of my model for this task of predicting breast cancer or for predicting lung cancer. Yeah?
Can you remove a location or some data? Do you remove it both from training and testing data, or only from training?
So for the Shapley value, we're just only removing it from the training set, right? So imagine that you have some external test data sets that we're not going to touch, right? So we're only just seeing how much this removing it from the training data set, which disjoint from the test, affects the performance. Yeah?
So here is the Shapley value associated with the location. The sum of the Shapley values of all the individuals.
OK, so good question. So here we're just computing explicitly for each location as a whole set what the Shapley value of that set is, which actually might be a little bit different from the sum of the Shapley value for the individuals. So I'll tell you in a bit, the next slide, of what is the value of the individuals. OK, so basically we want to figure out, right, so how does it happen that, how can this particular location's data be so negative and harm the performance of the model? Right? Maybe the first hypothesis you might have is that, OK, so is the data there just somehow noisier? Is there some technical effects? Right, so that turns out not to be the case to the best of our ability to tell. I mean, I think the data there is standard kind of QC metrics is actually quite high quality. So it does appear that the demographic distribution of that location, right, especially of individuals who end up developing sort of incident cases for lung cancer, looks quite different compared to the national average. Right, so if you compare like the age distributions of who are the people who develop lung cancer in this location 15, right, so that's actually different from the rest of the country. Right, so that's sort of our working hypothesis, is that there could actually be a large number of potentially outliers, either due to demographic reasons or due to potentially real disease mechanistic differences. Right, that makes data from that location quite different from the rest of the country, so that if I include that data in my model, which is sort of evaluated on the entire country, as it's commonly done, it's actually hurting the model's performance on the overall. So we tried to basically dive a little bit deeper into this. Right, so one way to dive deeper into this is, OK, so I actually have the Shapley value for each location, but also for each individual. Right, so now I can say, OK, so what if I just systematically remove individuals that have low or negative data value? How does that change the model's performance? As you can see, actually see on this sort of independent test set, right, a different test set from the one that was actually used to compute these values, right? So the performance model is actually improving as we're removing these individuals. Right? Whereas by contrast, if I just remove random individuals, that's the red line, or if I remove individuals that have small leave and out scores or small leverage or influence scores, you're actually dropping the performance of the model. So the flip side of this is we can also see what happens if we try to use this data Shapley value to try to recruit more individuals. Right? So maybe if I try to recruit more individuals that are similar to the ones that have high values, could I actually use this way to maybe to do active learning and to further improve my model? And we do see that it's actually recruiting high value individuals tends up to really significantly improve the performance of the model, much more than if I just recruit from the random population or recruit by using sort of the standard leverage score. And the last part is that, so there are many data sets where the data is actually quite noisy, right? And perhaps, as we said, you could have negative values if the data point is simply just very noisy, or if it's even maybe misclassified or mislabeled, right? So for very noisy data sets, we actually went back to explicitly check each data point to see is it actually misclassified or mislabeled, right? So here it's basically we're sorting the data points from the smallest negative Shapley value onward, right? We're checking each of these points, and we see that, okay, so actually a significant fraction of them that have low values are indeed the ones that are mislabeled, right? Much more so than expect if you just do random sampling of data points, or if you do sampling according to sort of leave one out estimators. Right, so we think that, okay, so the Shapley value is giving us quite a lot of information about the quality and heterogeneity of different types of data in my training set, and we can actually perhaps use this to improve my model. Okay, so what's the simplest idea to how to actually apply this to really improve the model, right? So we already see that maybe just even throwing away the data points, low value data points, can lead to some improvements. How do I do this more generally? So here's one example of this, just for give you another applications domain, right? But similar idea, right? So let's say if I built an image recognition system, right? Now it's typically trained on very standard benchmark data sets. And these benchmark data sets, actually quite well known, are quite much more heavily biased towards white males. And if you actually apply this image recognition system in practice, when it sees more diverse individuals, the performance suffers quite a lot. The performance drops. And now what we have is that we actually have a value, a Shapley value for each of my training image. Right? Some of them will have negative image, others will have positive image. Right? The simplest idea is that why don't we just simply weight every training point by its value, right? Just so that the model then will pay more attention to the ones that are likely to have more positive contributions. Right? So here's just a random collection of the images that end up having high value for this benchmark data set, and you see that they actually capture a lot more, tends to be a lot more female, much more diverse. Right? So then what we do is that we just take the exact same model that you used before, right? The exact same ConvNet. The only difference is that now it's trained on this weighted data set rather than the original unweighted data set. It's still trained on the top data set above. Right? And the performance, you can actually get quite a substantial improvement, about 8% improvement, when you deploy this model to more diverse individuals. A sort of a similar kinds of application is in this, for medical kind of imaging, clinical imaging. Right? So there's a lot of interest now in basically built AI systems that can take, like skin images, very noisy image, right, and can make, or at least for screening purpose, like make sort of predictions on whether is this benign or malignant. Should you be worried about this part of your skin? So the data set that's actually being used to train these models, it's extremely noisy, right? So basically, what they do is that they will go on Google Image and they'll just type in these keywords, right, for different skin illnesses and scrape images. Right, so that's how they get data sets of sites, hundreds, thousands, and millions data points. And when you actually apply them to real clinical examples, the performance is actually quite bad. It drops a lot. Right, so we basically do the same idea here. Right, now we actually have a value for each of my noisy training data points. The training images that are extremely noisy would have negative values or have very low values. Right, so the high value ones tends to capture images that are much more informative. right? So training the same model now on this image data set that's weighted, right, actually immediately gives double-digit improvements in the performance when it's actually applied to the clinical settings that we care about. All right, so we think that basically the message here is that thinking about the value of different data sets, right, in addition to certainly the policy implications, right, how do you basically compensate individuals, all that discussion, even just for the core statistical machine learning purpose is actually extremely important, right? Especially as you're getting data sets from heterogeneous sources, especially noisy labels, right? So this could be a way to quickly tell you what is the value and the quality of different types of data, and simply weighting the data by their value actually really substantially improves your model's performance. Great. So for the last couple of minutes, so we've also been, now that we've been developing these models and developing ways to improve the training data sets, right? And we really like to develop platforms that can make all of this very easily accessible, not just to the people in this room, but to your friends who are not even computational biologists or computer scientists, right? To really to the clinicians and to the biologists who do not even necessarily know how to program or how to, code in Python. So we've been trying to develop these frameworks, right? So it started off by making this sort of interactive tutorial, which I think is one of the first interactive papers that's published in Nature Genetics, where people can just go online and then really play with the model and get feedback in real time. And then more recently, we've been developing this framework that we call Gradio, which is essentially a way to make the sharing and accessibility of machine learning models, especially deep learning models, extremely easy for non-specialists who do not need to know how to program or how to code. Right, so the idea here is that, so let's say if you're a developer, right, so you write maybe some big Python package, you write your favorite TensorFlow model, right? So all that you need to do is to just add 2 lines of code to call this Gradio package that we wrote. Right? What Gradio does is that it's going to basically create an interactive wrapper around your algorithm, right? And it also creates a URL, a link, so that anyone with that link now can just open up the model on their browser without any download and just immediately interact with the model in real time. Right? So you can just create this link, send it to all of your clinical collaborators or biologists, and the Gradio just automatically creates this interactive user interface and so the front end so that your collaborators can just immediately play with the models in real time to make it extremely simple. Right, so your collaborators, when they open up the link, right, if you have an image recognition system, this is the kind of interface they will see. Right, so they can actually put it in their own images and then just play with it. Right, so I'm going to then try to do something quite dangerous, so I'll try to do a live demo of this to see, just give you an illustration of how this works. Okay, so this is basically Gradio, right? No, you can follow along if you want, right? So make this larger. Okay, and so there's like some simple tutorials, very easy to help you get started on this, right? So it's really just you import Gradio, and after you've built your model, it's writing these 2 lines of code, right? And Gradio will create different types of interfaces that allow for interactions with images, video, audio, even text data or genomics data, and you can customize your interface. So the other part of Gradio is that in addition to creating this tool for sharing the models, right? So we also basically create Gradio Hub, which is, think of that as GitHub, but now you can actually, GitHub is really for people who don't know anything about machine learning or models, right? So you can actually just immediately interact with any of these models on Gradio Hub through this interactive interface. Right, so we've basically uploaded, and other people can upload 2 different types of models. You can do it for medical imaging, for genomics, right? So people mentioned earlier, I think Elena mentioned earlier, DeepC, right? So you can actually interact with DeepC on this interactively. And people can upload these models, right? So when you actually open up one of these models, right? So for example, the first one, right? So you actually have sort of interface, like look at this, right? And let's say imagine that I'm just a practitioner. I don't know how this machine learning works, right? So what I would like to do is maybe I'll just try to grab some image that I have, right? And then just want to see, running this on my browser in real time, right? What actually happens to the model's prediction, right? Oh, okay, so this one actually works pretty well. And maybe what I like to do is to have some interactive ways to like perturb the image, right? Maybe I'll draw on it and see, okay, so how does the model actually change, right? So I don't actually know what's going to happen here. Oh, so it still got it correct, right? Maybe the confidence has dropped a little bit, right? So we create these different ways. You can also crop the image, right, to make it really hard, right? And then so this becomes a very easy way for practitioners, clinicians, to tinker with the model, right, and then see their performance. You can do this also with the skin cancer one that I mentioned before, right? You can apply this model. Okay, so we'll say it's malignant. Let's see what happens if I try to modify the model, because oftentimes you want to see by interacting with it, right? How these perturbations maybe are rotated. How robust is the model to these different, to your conditions, to your data. Right, okay, so here the accuracy drops a bit, but they're still pretty confident. So now you can actually just go on this, go on Gradle Hub, right? So if you're just interested in playing with these models, or if you have models that you would like to upload, right? So we can just make it very easy for people to play and to interact. You're on. Oh, okay, great. So that's actually a good time maybe for me to stop and wrap up. I mean, I just want to thank my students, especially, so the work with the CRISPR machine learning algorithm is mostly driven by Amir Ali. And then the data evaluation work is led by my PhD students, Atta, who's both of them have done really fantastic works. So happy to, maybe I'll just take some more questions.